and some people still coming in on Zoom, but we're going to get started. So today it is my pleasure to introduce the soon-to-be Dr. Sade Clayton Williams. Sade came to UAB in 2016, and she had graduated from the University of Georgia, where she had already won many scholarships and awards and already had a significant amount of research experience. I remember when she came for her interview and she interviewed with me, she was so excited about the research that she had been doing as an undergraduate that I actually got excited about it too, which, as you know, if you've interviewed a lot of students that are coming in, that, you know, sometimes those interviews can be kind of dry and painful. But when Sade left my lab, I was excited and energized about a project that I didn't really even know anything about. And so I was really pleased when she decided to come to UAB. And then when she decided to do a rotation and then join my lab, I felt very honored that she would come to my lab and work and bring her excitement and enthusiasm with her. And as you'll see today from my introduction and her talk that she's going to give, she's been a superstar since day one when she got here. And we'll all be very sad to see her leave, especially in my lab. So let's see if I can get this to work. Spacebar. Thank you. No, that's not working either. How do I make the slides go forward?
Click on the screen and fix
This and this. So this is Sade in 2016 when she came for her interview. You can see her excitement in that. It's right there. She's carrying the signs and which she goes. And then she joined my lab. She did the third rotation in my lab and joined my lab shortly thereafter. And I have her sitting here, but I can't read it because it's too small. So I'm just going to have to name it. So she joined my lab in 2017, and this was at a time before construction and before COVID, when we used to have lots of parties, lab parties. So this is one of our lab parties with lots of food when Sade had first joined the lab. So over her time at UAB, one of the things that Sade is very good at is winning awards. So she wins awards, and this is just a very small subset of some of the awards that she's won since she's been here. She's won the 2018 GPS Outstanding Mentorship and Teacher Award. She won a very prestigious poster presentation award at a Gordon Research Conference, and you know how hard it is to get into the conference alone, let alone win the award. And she's won several service awards at UAB, including the Blazer Way Service Award and the Ever Faithful 50 Service Award. So in addition to winning awards, Sade has been to a lot of meetings and has given oral presentations on her work based on her abstracts, on the quality of her abstracts. She's been selected for several talks, including local, regional, and national meetings, including most recently the Orthopedic Research Society 2021 annual meeting, where she talked about the work that she's going to discuss today. Now, not only does Sade go to these meetings and present her research, she also gets money to go to these meetings. She was awarded a scholarship for a stem cell and regeneration course. She's won several travel grants, including a travel grant to the Orthopedic Research Society and several travel awards from the Graduate Student Association. She's also, in addition to being able to find funding for her own meetings, she's been able to find funding for her stipend and her research. For most of the time she's been here, she's been funded on the CAMBAC training grant. So she's been on that T32 for several years. And for her postdoctoral fellowship, she was awarded Rita Levi-Montalcini Postdoctoral Research Fellowship at Washington University, where she'll be starting at, I believe, at the end of August or September 1st. She's had several publications including 2 first author publications. She has a first author review article, which I will say, thank goodness Sade was here because I would have never finished that review article. She pretty much wrote it and I read it and was like, yes. and then sent it in. Like, I think we made it by the deadline, which is very, very rare. And she also has a work that's in preparation that we'll probably actually submit next week or the week after that. She also does a lot of other like service and teaching and mentorship. She was awarded a Center for Integration Research Teaching and Learning Certificate, as well as a Teaching and Learning Associate Teaching Certificate. So she has, in addition to her normal GBS classes, she's taken extra classes in order to beef up her teaching skills. And this has shown through in some of her teaching activities that she's actually done at UAB. She's helped teach in some of the graduate level classes, and I know she's done a lecture for her alma mater in Georgia as well. She's also a very strong mentor. She's mentored students in the CIVS undergraduate program over the summer. She's a mentor in the Women in STEM committee, and also is a participant in the Girls in Science and Engineering Day Group Leader. And I also must stress that I've selected a very few from the pages and pages of teaching, mentorship and service that she has on her CV. And here's some example of her service and leadership. She's on the Orthopedic Research Society Spine Section Membership Committee. She's in the Equity Leadership Council. She was the president of the Graduate Student Government here at UAB for a while. She's on the Graduate Biomedical Sciences Curriculum Committee and has also been on the Admissions Committee. And anybody who's been on any of those committees knows how much work that is. And it really takes some dedication to do that, plus get all your research and other things done. So I would say that Sade is definitely ready to graduate. This is her in 2016, super excited, big smile on her face, ready to go. And look, 2021. Big smile on her face, super excited, still ready to go. I also want to tell everybody that we're going to have a reception over in Shelby 102. That'll be after the committee defense. So we're hoping that's around 4 p.m.-ish. We can all meet, you guys can meet over there at 4, even if we're not there, and get started on the cake and the champagne. But we will be there shortly thereafter. Everyone is invited. And so today, Sade is going to talk to us about TGFbeta provides the backbone of embryogenesis, elucidating the role of TGFbeta signaling and its downstream effectors on spinal column development. Okay, Sade.
Okay. Okay, so, wow, I was really excited to see that first picture. I've never seen that one before. I would like to know your sources. But thank you so much for that very kind introduction. I'm very excited to share with you guys what I've been doing over these past 5 years. And I'm very excited for your questions, especially for my second project. It is a work in progress, and so I'm excited to see what feedback will guys have for me. So Rose has already introduced the title of my talk, so I'll go ahead and transition to the next slide. So this is a brief introduction of what I'm going to talk about with you guys today. We're going to begin, of course, with TGFbeta signaling and the role that it has in spinal column formation. I'm then going to transition into the 2 projects that I have that encompass my dissertation. The first project talks about the mechanistic way that TGFbeta is regulating fibrous tissue differentiation within the axial skeleton. And the second project discussed a process called segmentation and the role that TGFbeta signaling is playing in that process to also regulate spinal column formation as well. So I'll pause after each one of these projects for you guys to ask any questions, and of course at the end you can ask any questions as well. So just for clarity, I hope that you will hold your questions till each project is done, so people want to have a chance to answer questions as well. Okay, so today, yeah.
Is there a way to do that?
Yeah, you are, yeah. I can do that.
May interfere with the pictures.
Okay, so today, like I alluded, that we're going to be talking about how the spinal column develops, but why is studying spinal column formation, why is that important? Well, when the tissues within the spinal column subject to degrade due to age or trauma, that can cause severe chronic back pain. So let me introduce you to the components of the spinal column and what I mean by spinal column. And so it is not the spinal cord, I get that all the time. The components that I'm talking about are the cushions of the spine, known as the intervertebral discs. From now on, I'm going to call that IED, just to save the time. And that's this white portion. Can you see that? Yeah, cool. That's that white portion here. And situated in between those intervertebral discs are the bones of the spine or the vertebra. And so what happens is that, again, as we age or experience trauma, those intervertebral discs can begin to degrade, compress on spinal nerves, and cause chronic low back pain. And as you can see from some of the stats that I put up here on the screen, is that it's a leading cause of disability worldwide, and it's very expensive. You can see that second statistic I put there. And this is an aging disease. And so most of us in this room, by the age of 50, around 86%, will experience or show some type of evidence of disc degeneration, and again, which can cause and lead to chronic severe back pain. Now, we're going to be focusing in on again the intervertebral disc. So let's look at those tissues a little bit more closely. So the intervertebral disc, again, that cushion of the spine, also consists of two salient parts. There is a nucleus pulposus, which is the inner structure that is gelatinous and is made to withstand compressive forces. Now, surrounding that gelatinous structure is a fibrocartilaginous or a fibrous structure called the annulus fibrosus, and that is made to withstand the tensile forces within the spine. Now, for this talk, I'm going to be focusing on fibrous tissues like the annulus fibrosus because as we age again or experience trauma, that fibrous tissue can begin to tear and degenerate, causing that nucleus pulposus to extrude out of its internal cavity, compress on the spinal nerve, and contribute to that severe chronic back pain like I just mentioned. Now, there are no disease modifying treatments to slow down or stop these degeneration of these fibrous tissues. Currently, the treatment is for pain. So doctors will provide or prescribe NSAIDs, maybe prescribe a little bit of physical therapy, epidural shocks when it becomes very severe. And the end all be all is spinal fusion, where you go into that affected region, take out that intervertebral disc, and fuse the two neighboring vertebral bodies, which does alleviate pain. However, it sometimes does not fully alleviate that pain, and the intervertebral disc flanking, that fused region, are now more prone to undergo degeneration because of that strain. And so there really is a need for a therapy to treat this disease and not just its symptom, which is pain. Now, so that brings in me. So the goal of my project is regeneration. So what I aim to do is study the developmental pathways that go into fibrous tissue development so that we can then use those pathways in order to stimulate and regenerate tissue so that we can treat the disease and not just the symptom of pain. Now, the Sarah Lab has made great strides in determining the pathways that are important for this process by bringing in TGFbeta and the importance that TGFbeta has in spinal column development. Now, the spinal column develops from one single progenitor tissue, which is the sclerotome. And the sclerotome actually comes from a transient embryonic structure called the somite. And so, as shown here in yellow, now if you were to zoom in on the somite, after it's gone a little bit of differentiation, it turns into three structures or three tissues, which is the myotome, the dermatome, and the sclerotome. Now that progenitor tissue that turns into or develops into the spinal column tissues, the sclerotome, can go down two different cell fate decisions. It can either go down chondrogenesis to make cartilage, to make the vertebra of the spine, or it can go down fibrogenesis, or is that? Okay, perfect. So, or it can go down fibrogenesis to make the fibrous tissues of the spine. We've seen annulus fibrosis in the previous slides, but fibrogenesis can also make tendons and ligaments, other fibrous tissues. Now, our lab has shown that TGFbeta acts as a master regulator during this process because, I'll just leave that one, I'll get to that one next. TGF-beta has the capacity to inhibit chondrogenesis, which goes to make bone, via chondrogenesis, making cartilage, then ossifying and making the bone grow. We also have shown that TGF-beta acts as this regulator of fibrogenesis that regulates and pushes these cells toward fibrous tissue differentiation by causing expression of these fibrous tissue markers. 3 of those are listed here. Do not worry, I'm going to get very deep into that in the first project. But we've also shown that we've also shown that without TGF-beta signaling, by deleting TGF-beta receptor 2 in these cells that go on to form these tissues, that the annulus fibrosis is missing, and the vertebra, the bones, are malformed. And the last thing that other labs have shown is that if you delete 2 of the TGF-beta ligands, TGF-beta 1 and 3, that tendons and ligaments throughout the body are missing. And so all of these studies, not only from our lab, but from other labs, show that TGF-beta is incredibly important for development of the tissues of the spinal column, with a special emphasis on fibrous tissue differentiation. Now, I wanted to know how is TGF-beta doing that? What is the mechanistic way that TGF-beta is regulating these processes? So in order to address that question, first I'm going to get you accustomed to TGF-beta signaling within these cells. Now, TGF-beta acts as a homodimer that binds to a heterotetrameric complex at the cell membrane of these sclerotomal cells that consist of 2 TGF-beta receptor 2s and 2 TGF-beta receptor 1s. Accumulation and formation of this heterotetrameric complex causes activation of the activation domain on receptor 1, which causes activation of downstream effectors. Now, TGF-beta can signal in 2 flavors, if you will. It can undergo canonical signaling by signaling through SNADs, or it can undergo non-canonical signaling by signaling through effectors like AKT, P38, as well as ERK1 and 2. Now, all of these effectors, the end result is that they activate downstream targets, which it turns on specific gene transcription, targets and causes different effects. In this case, fibrous tissue differentiation. Now, pay attention to the ones that are highlighted in red. You will see them again, they are important. And so I wanted to determine which of these pathways are important for TGF-beta to signal in these cells to stimulate fibrous tissue differentiation. Okay, so how am I going to test this? Well, for this first project, I use the mouse model. What I do is that I take E11.5 mouse embryos, isolate the spine, as outlined here in this black box. I then isolate the sclerotome from the spine, shown here in blue. And what I do with that sclerotome is I then plate them under micromass conditions. Now, this is a condition which has a high density of cells, about 300,000 cells per 20 microliters of media. And this is just to stimulate differentiation. So these are these little balls, those are the micromasses. And what I do is I treat those micromasses with TGF-beta for 2 or 8 hours, and then use Western blot analyses and qPCR to look at fibrous tissue differentiation markers. That came up too early, but it's okay, we're going with it. So on the flip side, in order to test which pathways TGF-beta is utilizing, I use inhibitors. And so in this case, I treat the cells with an inhibitor first for 24 hours, and then similarly, I treat those cells with TGF-beta for 8 hours afterwards, and then I'll look at Western blot and qPCR to look at fibrous tissue differentiation analysis. Now let's get into some data. Okay. So the first thing I wanted to show was that TGF-beta was regulating these fibrous tissue markers in this system, and this system is the primary sclerotome I isolated at E11.5. So what I'm looking at here is the relative RNA expression. This is qPCR analysis, and what I'm doing is I'm treating these cells with nothing or a vehicle, or TGF-beta 1 ligand for 2 or 8 hours. So what you can see is that I have 3 markers listed here. These are the fibrous tissue markers I'll be using for the rest of this talk to denote that fibrous tissue differentiation is happening. Sclerosis is a transcription factor well known to regulate tendon development of fibrous tissue. And these other 2 markers are fibromodulating additives, so 2 they are extracellular matrix markers known to be enriched within fibrous tissues. Now, what you can see from this data is that this second marker gives control is that only sclerosis is regulated by TGF-beta treatment at 2 hours, and it takes up to 8 hours of treatment to finally regulate F-blot and anti-TSL2. And because of this, I put these factors into two categories. I call slow access the earlier regulated marker, while these are the two later expressed marker. So it's showing the temporal fashion that TGF-beta regulates these markers. But I wanted to know, did TGF-beta need protein translation in order to regulate these markers? To do that, I treated cells first with cyclohexamide and then with TGF-beta, then to determine does TGF-beta regulate these markers in a direct or kind of indirect fashion. So first we looked at sclerosis. So these first two bars are controlled. So without and with TGF-beta, we see sclerosis RNA is regulated. However, these last two bars are treatment with cyclohexamide alone or treatment with cyclohexamide in the presence of TGF-beta. And what I see is that even when new protein translation is being inhibited, TGF-beta still has the capacity to upregulate sclerosis RNA, indicating that there is a somewhat direct fashion that TGF-beta is regulating this marker. Now, what about the later expressed fibrous tissue markers? What I see is that that's not quite the case. So again, the first two bars are controls, TGF-beta upregulates this RNA. However, if you compare cyclohexamide treatment alone to cyclohexamide in the presence of TGF-beta, for both markers, you see that TGF-beta can no longer upregulate these markers, showing that TGF-beta requires protein translation, indicating an indirect way that TGF-beta is regulating these markers. So this first panel is to show that there's different ways temporally that TGF-beta regulates these markers, and that seemingly direct and indirect fashion that TGF-beta could be regulating these markers. Okay, so we're showing again. The purpose of this project is to get into the way that TGF-beta in terms of canonical or non-canonical pathways that TGF-beta is regulating everything. And so the next thing I wanted to look at was canonical signaling. How is TGF-beta regulating this? None of my animations are working, so stay with me here. So we're first going to focus in on this western blot here to look at phosphorylation or activation of SNAP3. We treated these cells with TGF-beta for 2, 8, and 24 hours. So you can see that TGF-beta does regulate phosphorylation of SNAP3, but not total SNAP3 over these different time points. And this is quantified here with densitometry analysis to show that phosphorylation over total SNAP3 accumulation. And that is statistically significant in 2 and 8 hours. So we showed that TGF-beta activates SNAP. Wow. Okay, now it's okay. So we showed that TGF-beta activates SNAP3 at these different time points. Now we wanted to test is SNAP3 required for TGF-beta mediated regulation of these markers? And to do that, we treated these cells with SIS3, a SNAP3 inhibitor, at 2 different concentrations. And looked at the RNA of these markers again. Now, PRT4 is a positive control of SNAP3 signaling, and that's why that's there. So if you look at these markers, what you can see is that if compared to control TGF-beta treatment alone, that sclerosis, if you treat with this inhibitor, there's no change, indicating that TGF-beta does not require SNAP3 to regulate sclerosis. However, these later expressed markers of F5 and TSO2, with increasing concentrations of SNAP3 inhibitor, TGF-beta can no longer regulate these markers to the level that it was regulated before, showing that SNAP3 is required for the regulation of these later expressed markers. And again, PRT4 is that positive control. Now, so that shows the necessity of SNAP3 in this process. What about is SNAP3 sufficient? Is it sufficient to regulate these markers? Now, to do that, I overexpress SNAP3 to an adenovirus and use a GFP adenovirus as a control. So what I can show you here is that when we first look at this positive control of SNAP3 signaling PRT4, if you compare the first and the third bars, so this is the control bars without and with TGF-beta, you see that TGF-beta signaling regulates this marker, well established in the field. But if you compare the addition of SNAP3 in the presence of TGF-beta, you can now see that this marker becomes super induced. I'm showing that SNAP3 cooperates with TGF-beta signaling to further regulate this marker. Now, with that in mind, let's look at these fibrous tissue markers. What you can see at the first and third bars is that, again, TGF-beta regulates these markers, well established. Now, if we look at the last 2 markers, last 2 bars, what you can see that TGF-beta does not have, or SNAP3 does not have the capacity to super induce these markers as it did with this positive control, showing that TGF-beta does not need SNAP3, it's not sufficient to regulate these markers. So this basically establishes that SNAP3 is required, at least for these later expressed markers, but not sufficient to regulate these markers. So that tells me that what I'm not looking at is the whole picture. TGFbeta has to be used some other effectors to regulate these markers. And so that made me want to look at non-canonical ways. That made me want to look at non-canonical ways that TGFbeta was using to regulate fibrous tissue differentiation in the scleroderma at this time. So, for the sake of time, I'm going to get to my second project as well. We looked at all the markers that I showed you. We looked at PhD, we looked at AKT, and those non-canonical, we looked at those pathways because they have been indicated that they were required for skeletal development by treating them with inhibitors and seeing if TGFbeta could still regulate these markers. We showed that it can. So they were not required for TGFbeta mediated regulation. However, ERK was. And so let me go ahead and show you that basically for each one of these non-canonical pathways, ERK, P38, and AKT, that at 2, 8, and 24 hours with treatment of TGFbeta1, they were regulated when you compare them to their total controls. And that was a statistical difference at 2 and 8 and 24 hours for ERK, and at 2 and 8 hours for PAKT. But like I mentioned, only ERK was required. Why? But only ERK was required. I apologize. I don't know here. Okay, so in order to test that, again, we use these inhibitors to inhibit its activation. And in this case, this is a MEK inhibitor. MEK is the kinase directly upstream of ERK that activates it. So we use 3 different concentrations of this MEK inhibitor. So if you compare TGFbeta treatment, where you see there's an activation of ERK, with this increasing concentration, there's a downregulation of ERK activation that is quantified here with, again, this photometry analysis. Now let's look at its effect on fibrous tissue marker expression. So what you can see from here is, again, with the control TGFbeta treatment alone, but with increasing concentration of this ERK inhibitor, none of these fibrous tissue markers are able to be regulated by TGFbeta in the same fashion. And again, this shows that ERK is that non-canonical factor that regulates all of these markers, even sclerosis, which SNAP3 is just not capable of doing. And this again shows that both canonical and non-canonical factors work together in terms of TGFbeta signaling to regulate fibrous tissue markers and therefore fibrous tissue differentiation during this process. Now, I keep mentioning how different these markers are. That sclerosis is regulated first, and that it seems to be regulated in a direct fashion versus an indirect fashion. I also mentioned that sclerosis is a transcription factor and a well-known tendon regulator within the tendon field. So I wanted to test, could sclerosis also be playing a role in regulation of these later expressed markers, since it is itself a transcription factor. So to test this, I use C through H10 T1/2 cells. These are very similar. Immortalized cell lines similar to primary scleroderma cells. I treated these cells with a sclerosis siRNA in a controlled scrambled siRNA. I saw that the RNA was downregulated with treatment of this sclerosis-specific siRNA. I then checked for protein, saw that the protein compared to control was downregulated, and this is the quantification of that protein here, so that it is statistically significant. Now let's look at its effect on these factors, FMOD, later expressed factors, FMOD and TSL2. So the 2 gray bars here are the control, the scrambled siRNA without and with TGFbeta treatment. You see for FMOD and added TSL2, again, TGFbeta works, I've shown you many times. However, let's look at what the presence of this sclerosis siRNA does. So if you compare without TGFbeta treatment to with TGFbeta treatment for FMOD and added TSL2, you see that even in the presence of TGFbeta, these markers are not regulated anymore. And this shows that their TGFbeta is, that sclerosis is a required factor that is necessary for TGFbeta's regulation of these markers, because if it wasn't, these bars would be upregulated because TGFbeta is present. So interestingly, not only sclerosis also plays a role in the basal expression of FMOD. Because if you look here with scrambled siRNA and sclerosis RNA with no presence of TGFbeta, there's a downregulation in the basal expression of FMOD. So sclerosis not only plays a role in TGFbeta mediated regulation in both markers, but also the basal regulation of FMOD in this process. Now that was a lot. So let's bring it all home. Okay, let's bring it all home with a model. So what I think is happening So what I think is happening during this process is that in the sclerotomal cells, TGF-beta signaling in this process activates ERK first. And this activation of ERK turns on fluorescence RNA. Now that RNA is eventually turned into protein, and I think that in collaboration or cooperation with SNAP3 activation by TGF-beta, that these two effectors work together to turn on ADAMTSL2 F9 gene or RNA regulation, which again are fibrous tissue markers, and this stimulates fibrous tissue differentiation. Now, so this is a novel pathway. This is a pathway that shows that both canonical and canonical effectors work together to mediate the process of fibrous tissue differentiation. And I believe that we are likely looking at a snapshot of what's happening here, and that these markers likely work in conjunction of complex webs of other, factors to stimulate the different types of fibrous tissues throughout the spinal column. So there are multiple remaining questions. That's how it's looked at this project, which is how is TGF-beta signal regulating scleraxis? And do scleraxis and SNAP just cooperate, or do they interact directly together to regulate these downstream targets? As well as how this ERK regulating fibrous tissue markers, it is a kinase and a transcription factor. So what is it directly targeting to regulate these processes? And the implications of this project is that it can be used in regenerative therapies to treat degenerated fibrous tissues. And I envision this as a part of a kind of stem cell therapy in order to regenerate or repair these tissues. Now, this is the end of the first portion of this, so I'll take any questions that you have so far. That was a lot. Go ahead, David.
So this MET kinase inhibitor, does it influence or does it impair development of embryos in the murine model or any model? So it can treat an animal during gestation to get some effects.
So this specific inhibitor itself. So I'm not sure. Again, I took these cells out of the embryo, these sclerotomal cells, and treated them in an in vitro setting. However, since what's downstream of this is ERK signaling, it has been shown that alterations in ERK1 and 2 causes kyphotic, spinal defects. And so your kyphotic defects are the thoracic version, the real version of the spine. So that has been shown to be happening.
Is that MET inhibitor used in humans for some purpose, or is it just for compound?
I'm not sure about that. Thank you.
Do you know the source of the TGF-beta? Because you're not isolating the primary sclerotome from a knockout or anything, right? You're taking them out and then treating them. Is there some innate TGF-beta already in there that's coming from any of those cells, or is it coming from other?
Yeah, that's the old faithful question that I get every single time.
Sorry.
No, it's fine. I love answering it because it's the same. We're not sure. I think that it's so uncommon is that these cells already are fine and expressing TGF-beta because we know for certain that they ubiquitously express the receptors for it.
So when
No.
you're treating, it's
Yeah.
almost like an overexpression? Is
Yes,
that the idea?
absolutely.
Okay.
Yeah. So we would love for someone to tell us what turns on TGF-beta, but we just don't.
Educate me a little bit about the first figure, figure 2 we put in the thesis about MSA differentiation in the fibrogenesis chondrogenesis. You say that TGF-beta regulates by non-canonical and canonical signaling for fibrogenesis
Yes.
differentiation, and at the same time it blocks also chondrogenesis,
Yes.
right? For differentiation of chondrogenesis also you need TGF-beta,
Yes.
right?
Yeah.
How does
So
it differentiate to stop it there, activate the fibrogenesis pathway?
so you're asking how does TGF-beta signaling specifically inhibit chondrogenesis?
Yes.
Yes, that was the previous, our previous graduate student, Joy, that was like her whole project, showing that, TGF-beta regulates noggin, known DMP agonists, that helps inhibit chondrogenesis. So that's one definite known pathway. Yeah, so that's the main one that I can think of right now. But yes, it seems to do both at the same time, inhibit chondrogenesis and then stimulate, because inhibiting chondrogenesis isn't enough. Inhibiting those cells alone, they just kind of sit there. They don't seem morphology wise or marker wise to be stimulated to be doing anything else. You have to apply more TGF-beta to stimulate them to go down fibrogenesis.
Sade?
Yes.
Hi, here from Athens. Hey, so I just have a question about the dynamics of the TGF-beta receptors activating both. Can you hear me okay?
Yeah, I can.
Okay, the dynamics of the TGF-beta receptors activating both the canonical and non-canonical pathways at the same time. When they go through the non-canonical pathway, are the receptors acting directly on ERK, or are they going through an intermediate like RAS? How does that work? And do you think the same complexes are activating both pathways, or are there different complexes that are dedicated to one pathway or another?
So I think that there are different things happening for both canonical and non-canonical. So studies have shown that with canonical signaling, that TGF-beta directly activates those canonical effectors. That it directly activates, a specific activation region within like the C terminal of these SMADs, and so there is direct. However, with non-canonical signaling, it activates other intermediates, and it's kind of like a downstream effect of what's happening here. Now, those intermediates change from cell type to cell type. I don't exactly know what's happening here in this system, but yes, it is different.
And within one cell type, would you have one receptor complex activating the canonical pathway and another receptor complex activating the non-canonical pathway, or are they just activating both complexes at the same time?
So the receptor complex itself consists, always of two TGF-beta type 2 receptors and 2 TGF-beta type 1 receptors. Now those type 1s can fluctuate. It can be like ALK 4, 5, or 7. However, it could be, typically I would think that it would be the same type of paratetramer complex acting on these 2 different pathways to stimulate this effect. However, science is weird. It could be activating, different versions of paratetramer complexes within these cells, but I don't think so.
There's a question in the chat from Lauren Boris. Do you plan on conducting future research to address the remaining questions about how SCX and SMAD3 interact to regulate downstream targets? If so, what are the next steps that need to be taken to answer this question?
So I personally do not. However, the Serum Lab is already, in process and underway and looking at those types of things. And that is well fleshed out in her brain. And so I don't, but yes, we do plan on doing that. And you said the interaction between SLORAX and SMAD3, correct?
Yes.
Yeah. So there's different types of, mechanisms and and experiments that can be done to test if it's, indirect, knocking out one, keeping one there. Doing co-IP to see if they're directly interacting. Many different things that can be done, and I look forward to reading those people's publications. Okay.
Yes, more on
One
this.
more. Okay.
Did you test if you did the siRNAs or axis? Do you still get phosphorylation of SMAD3?
So we have not tested the effect on, one effector on the other yet. So I did not look to see, if inhibition of SLORAXes affects phosphorylation of SMAD3. I haven't tested that.
I think that's maybe what they were asking earlier. Can you have one pathway that does a cell always activate both pathways, or can you have a cell that does non-canonical only and another cell that does canonical only, or are they both always active?
That's a good question. I would think that, if SMAD is there and these effectors are there and the receptor complex is activated, that they would just be, far away, that these things would be happening simultaneously. I have not tested whether that's actually true or not. Okay, let's move on, because I have a whole new project. Okay, so again, that was looking at the mechanistic way that TGF-beta is regulating this. Now I wanted to look at a process called resegmentation. And so resegmentation is honestly not a well-known process, but it's incredibly important. And I hope that I convince you of that by the end of this call. So before I introduce to you what resegmentation is, let me introduce to you what segmentation is of the embryo, because I recognize a lot of us are not developmental biologists here, so I want to get us all in the same time. Now, segmentation is the production or the arrival of somites in the anterior to posterior axis of the embryo. So again, somites are these transient embryonic structures, and segmentation is the temporal fashion in the way that these somites arrive from a unsegmented tissue, the pre-Semitic mesoderm that's more posterior here, and the somites form in an anterior to posterior fashion. This is what it looks like within a chick embryo. So I wanted to know could TGFbeta be regulating this process of resegmentation? Now, again, what is resegmentation? Resegmentation is the reorganization of segmentation. And so essentially what happens is that it's the formation of rostral and caudal domains within the sclerotome, a derivative, a tissue derivative of the somite. And these rostral and caudal domains actually separate from each other, recombine with their neighbor, and form a whole new sclerotomal segment. And so what happens is that if you were to label somites within the a chick embryo, different colors, their tissue derivatives would be labeled those colors as well. But only and specifically in the sclerotome will there form these rostral and caudal domains. And these rostral and caudal domains will, as I said, separate from each other and recombine with the domain in the neighboring sclerotomal segment. And this whole new segment is where the vertebrae will form. And that in-between region, that border region called von Eggman's fissure, is where the intervertebral disc will form. And this is thought to aid in the spatial organization of the spinal column. It's also shown to be important, especially the formation of these rostral and caudal boundaries, because the spinal cord nerves egressing from the spinal cord have to cross the sclerotome to reach the myotome on the other side and innervate them. And it's been shown that they specifically only cross the rostral region of the sclerotome. And if you were to delete these rostral and caudal domains, they could not cross the sclerotome. So resegmentation is incredibly important for the organization of the spine. Now, I wanted to know, could TGFbeta be playing a role in this process, because disruption or resegmentation has effects on spinal column development that deleting TGFbeta signaling has as well. So if you look at a wild type, in this case, this is mouse. If you look at a wild type spine and you stain for fibromodulin, a fibrous tissue marker that's labeled specifically the IVDs, you'll see that there's this metameric segmented pattern of the IVD. However, if you caudalize the spine, meaning that you knock out those rostral markers, in this case, NUS2, so only caudal markers are expressed throughout the spine, there are severe spinal defects. So for instance, specifically in the lumbar region, these IVDs are completely fused on the ventral side, and the middle portion of the IVDs are completely missing in some of these regions. And if you rostralize the spine, the opposite, delete all the caudal markers, and so that only rostral markers are expressed, it's even more severe. So what you can see here is that there are complete fusions, dorsal and ventral to the spine, and that in the lumbar regions, a lot of the IVDs are almost completely missing. So resegmentation shows a lot of the severe phenotypes that we saw with TGFbeta deletion as well. And so that led me to the hypothesis that these processes may be working together, or maybe TGFbeta is regulating segmentation to aid in spinal column development. Now to test this, I'll be using the cheat model because it's awesome, and because it is a powerful developmental biology model where you can manipulate the embryo outside of the egg. And so what I do here is I take E2.5 chick embryos. I then inject them with a vehicle or this TGFbeta receptor 1 inhibitor, SB4B1542. From now I'm going to call it SB at the same time. And then I look at whole mount in situ of 24 after 24 hours of injections in these chicks to see if there's an effect on spinal column formation and TGFbeta targets. So in this case, I use scleraxis. So this is whole mount in situ using a scleraxis RNA code. So these arrows point to the border region outside of the injections, and inside what you can see is, if you compare the outside region, which is the control, there's this segmented pattern. These are the IVDs. However, where the injections are, those IVDs are just not as deeply stained with scleraxis, and that there seems to be a spinal malformation in this region as well. If I isolate the tissue from this injected region and look at scleraxis protein expression via Western blot, what I see is that in response to this TGFbeta inhibitor, scleraxis protein is downregulated, and that is quantified here in this differential analysis. So this shows that in the chick model as well, TGFbeta signaling is important for spinal column formation. So now let's look at the effect of TGFbeta signaling on this embryonic process of resegmentation. So what I do is something similar as what I've shown you. In the thoracic region of the spine, I inject somites 21 through 26 with alternating colors. This is so I can monitor what sclerotomal cell populations have incorporated into specific spinal column structures. Those somites that I mentioned turn into the sclerotome, and I can monitor how the sclerotome incorporates into the mature spinal column to determine if resegmentation is happening. Now, if it's happened, these colors, these labeled sclerotonal populations will incorporate equally from different sclerotonal units into one vertebra. And I can monitor resegmentation has happened because I see a vertebra labeled with 2 different colors. Does that make sense? Okay, cool. So let's see that happening. So I determined that resegmentation, can somebody get the lights?
That's great, yeah.
So I determined that resegmentation happens at embryonic day 6.5. And so what I did was I injected in each chick at E2.5. Now I put the mouse embryonic bone to egg. I realized nobody studies chick with me. So this is around E11 in the mouse. And what you can see is that this is the same day of injections that the somites are still intact. These are literally balls of mesenchymal stem cells, and you can see those labeled here in individual somites. Now 24 hours later, what you can see is that those somites have dispersed, undergone EMT, and are in their respective derivative tissues. Sclerotome and dermamyotome. There hasn't been much dorsal to ventral migration, which is what's required to form the spinal column. However, by E4.5, there's been extensive dynamic movement dorsally to ventrally, which is right to left, and top to bottom is anterior to posteriorly. So you can start to see that there has been a great expansion of growth in the embryo and a great labeled cells migrating dorsally to ventrally. 24 more hours later at E5.5, you can see that there is not only again extensive dorsal to ventral growth, but laterally, anterior to posteriorly, there's been a great migration and separation of these labeled sclerotonal populations. And most notably, you can begin to see a segmented pattern here ventrally, which is where the spinal column will form. Finally, about E6.5, I showed that resegmentation was happening. Now I showed that you can look at these blue brackets here. What those are outlining are the rib heads, where the rib comes out of the vertebra and eventually surrounds the ventral portion of the embryo. Those rib heads, which are cartilaginous, are half green, half red, or half red and half green. So this indicates that resegmentation is happening. Now in this view, these are all confocal volumetric images or maximum intensity projections. So about 200 micrometers of depth smushed all into one. So I could not see the vertebra here because actually these long fibrous-like cells are muscle and they're covering the vertebra and they don't undergo resegmentation. So in order to see resegmentation at the vertebral level, I took these embryos, sectioned them, and then I have the vertebra outlined here in white. What you can see here is that the vertebrae are indeed half blue, half red, indicate resegmentation does in fact happen at this day. So the purpose of this was to show that from injections at E2.5 to E6.5, resegmentation happens at this day. So I've shown you the day that it happens. Now let's see the effect that TGFbeta inhibition has in this process. So what you see here is a control or vehicle treated embryo at E6.5. What I have staying here is PNA, voice is the DNA stain. PNA is it's a lectin that stains diffusely throughout the spinal column, and I use it to outline the vertebral regions. And the yellow dotted line is the border. So if I then superimpose it here onto the labeled sclerotonal populations. And so these dyes, I'm so sorry I forgot to mention, are DID and DIO. They are lipophilic fluorescent tracers. They interpolate, and incorporate into the cell membrane of these cells, and so they're great lineage tracers because of that. So what you can see in wild type or control labeled embryos is that these labeled sclerotonal populations incorporate into vertebrae equally, half green, half red, half red, half green, half red. Now, how does that change, or does it change with TGFbeta inhibition? So yes, it does. We're on our project. So what you can see here is that incorporation of these sclerotonal populations are no longer equal if they begin to shift. And I put them into 3 different categories. So there can either be a full shift, where now a label or an outline vertebra only has 1 sclerotonal population, instead of that equal incorporation into 2. It can be a partial shift, where you can see in this outline vertebra here, that the caudal population, this green, has expanded at the expense of this rostral population. Or there could be a slanted shift, where still this caudal population has expanded, but now the border between these two populations is slanted. Or there could be a mixture of two, which is what is shown here. And so this shows that resegmentation is altered because resegmentation in itself is the incorporation of these labeled populations equally to make the vertebra. And so what I did is quantify that. So gray is rostral, or the top color, black is caudal, or the bottom color. You can see that in the control that they are equally incorporated. However, in the treated, the caudal is expanded at the expense of the rostral. And again, that can happen in different ways. So TGFbeta does in fact inhibit resegmentation. Now, again, it's been shown in the literature that these borders or these boundaries, rostral and caudal boundaries, correlate with the expression of specific markers within these boundaries as well. So some common rostral ones are like TGF18, that's 2. Common caudal ones are UNCX, I always get the X and the T mixed up, but 4.1, PAX1 and 9. So I wanted to see, are these rostral and caudal markers also disrupted? So this is an earlier time in the chick. This is E3.5, where the sclerotone is still present. Again, those other experiments were E6.5. What I first wanted to look at is a rostral marker and determine between the control and the treated, is these rostral caudal boundaries, are they still maintained? So for tenacin, I used it as a rostral marker here. What you can see is that in DMSO, there is a gaping hole here with tenacin is not expressed caudally, and that pretty much that's the same here in treated. So hoist again is I'm using that in the DNXA, and if you merge the two images, you see that again, tenacin is contained to the rostral domain of both the untreated and the treated. Now, what about the caudal marker? So in this case, I use again PNA because at this stage, it has been shown in the chick, it is restricted to the caudal region of the sclerotone. So if you compare non-treated and treated, you can see again here in the non-treated, it's restricted to caudal. However, in the treated, now there seems to be an expansion of this marker throughout the rostral and the caudal regions. Again, this is squished, and then the comparison, you can see again, there's a hole here, rostrally and the non-treated. However, in the treated, in response to TGFbeta inhibition, this caudal marker, in terms of PNA, has now been expanded. And so what that means is that there in TGFbeta inhibited embryos, there's the co-expression of these rostral and caudal markers in the rostral region where they should not be happening. These markers usually are transcription factors that stimulate different pathways. And I think the co-expression of these markers can be causing some signaling or differentiation defects that leads to spinal column deformity, which I show here. Okay. So in response to TGFbeta inhibition, this spinal column is altered. So this is E6.5 going back, later in development. This is untreated to treated. And what I show, if you zoom in here in these outlined black boxes, is that the vertebra, so this is Austrian blue staining, is that the vertebra, the walls are thinner in the treated, and that intervertebral disc space is reduced. And so that is quantified here with V wall thickness in E and average disc height in F. Now, I also show even further in development at E12.5 that not only is the intervertebral disc space reduced, but the IVDs are now formed. And I quantify that here in the area is that is reduced in the treated. And so this shows that multiple things are happening. Resegmentation is altered, rostral caudal polarity is disrupted, and overall the spinal column formation is disrupted in response to TGFbeta signaling. So the last thing that I really observed is that the muscles or the muscle fibers themselves seem to be thicker in TGFbeta treated embryos. And I wanted to quantify that and see if that phenomenon was actually true. So I used MF20, which is a myosin 2 marker. It's marking myosin 2 within these tissues. So if you compare untreated to treated, what you can observe in this zoomed in box here in white shown in B and C is that if you compare untreated to treated, TGFbeta inhibited fibers, they are much thicker than TGFbeta inhibited, and that quantification is here. Now that didn't surprise me when I looked at the literature. TGFbeta has been shown to be an inhibitor of myofiber differentiation usually, and so if you inhibit the inhibitor, that stimulates myofiber differentiation, and I observed that as well. And didn't get the chance to show you a lot of cool videos. I'm going to take that time to do that now. I also do a lot of live cell imaging of this process. So from E2.5 all the way to E6.5, I monitor what's happening at each embryonic day. We'll be here all day. So I wanted to show you that I saw this difference in myofiber. Oh, I hope it shows. Oh.
No.
I saw this. How do we do this? So I saw this difference in myofiber differentiation. And that it was evident even from using live cell imaging that these myofibers were, oh, thank you, here it is. So that these myofibers were different. So if you look at untreated, what you can see here, this is an E4, please. This is an E4.5 embryo. This is 12 hours of continuous imaging that has been sped up. What you can see is the popping into spring, these long elongated fibers in these different, labeled sclerodermal or somite populations. But if you compare that to a SB treated, one thing that you can definitely notice is that is still happening, but at a exacerbated amount. These fibers are more, and they're popping up. It's beautiful. It's more, they're thicker. And so that also led me to quantify this result as well, and to narrow down that these are in fact muscle fibers. Okay. So let's bring it all home. Okay, so I was forgetting a lot. So what I show here is that TGF-beta signaling under normal endogenous conditions. There is the establishment of this rostral-caudal polarity through the sclerotome, and that leads to normal resegmentation in the spinal column and a perfectly happy, normal functioning spinal column. However,
Shall
oh,
the Zoom is not showing.
it's not showing. Oh no.
Oh, it's still on the other.
It is.
Yeah.
That is weird. Okay, is it showing us now? Sorry guys, she was cool videos, but believe me, the myofibers were thicker. The model here is that again under normal endogenous conditions, TGF-beta signaling, there is normal rostral-caudal polarity that leads to normal resegmentation and formation of a perfect spinal column. However, when TGF-beta signaling is deleted or inhibited, what happens is there is a disruption in this rostral-caudal polarity where the caudal boundary seems to be expanded, and there's this co-expression of rostral and caudal markers within the rostral region. I think that this leads to kind of a self-identity crisis where there is probably differences in differentiation and signaling pathways, and also causing these cells to group differently. And this altered sclerodermal cell grouping leads to altered segmentation, since there's a shift in the way that these cells incorporate into the spinal column, leading to altered segmentation. And that manifests in a spinal column that is really funky. The BBs are messed up, the AFs are thinner and not formed. And so overall, this is how I think TGF-beta is really contributing to this very complicated process. So the novelty of this is that I'm the first to establish that TGF-beta is involved in this pathway, and that again there are multiple effects that happen. But again, as always, there are remaining questions. For instance, specifically is it canonical, non-canonical? How is TGF-beta regulating this process of rostral-caudal polarity? How does disrupting rostral-caudal polarity specifically affect segmentation? Again, I postulated how that can happen, but I didn't show any definitive ways. And our last question is, how is altered segmentation causing these spinal deformities? And I hope that this data can be used to treat resegmentation related disorders such as type 1 syndrome that causes fusion within the cervical spine and short stature. Okay, so with that, I would like to thank my lab, past members, especially Joy. Without her, I would not know anything chick related. And my thesis committee, and I'll take any lasting questions for this project or the one before.
A technical question. You obviously have the proper controls with your somite injections, but that is so early. Where exactly are you injecting, and is there any effect of the mechanical? Again, obviously you have your DMSO control, but if you were comparing it to one where you weren't injecting anything, is that different? Because it seems like that mechanical stress would have to mess stuff up.
Yeah, so the first part of your talk was like, where am I injecting? So I always know exactly where I'm injecting, so I'll use this messed up, this is actually a twins, that I took a
Oh,
picture of.
wow.
But this large blood vessel here is called the middle artery, and it crosses over the membrane at the exact same area in every embryo. And it crosses over the membrane at somite 23. So I use that somite and I inject around it to inject the same place every single time. I ask for mechanical stress. Is there a difference? I have not seen it. So what I do is that I culture, the eggs the regular way in their yolk happy, and then I isolate them out of the yolk, put them in this artificial system, and I culture them at the same time. Take images of them, also look at morphology to see if there's a difference, and there doesn't seem to be. Yeah.
So in your videos, you showed your DMSO injector control and your inhibitor injected. And in the inhibitor injected, it looked like there were a lot of yellow cells that were like co-localized, red and green together. Can you speculate as to what those are and if there are actually more of them in your inhibited versus your control?
Yeah, so what those are is, so even the videos again, when I take them, I make maximum intensity projections of them. So again, it's all volumes smashed together. And so those co-localized ones are very superficial. So I think it's from where the spillage has come out, we just firing away at somites. And then at the superficial levels, the dye might mix. But it has been shown, and even I see that at the deeper levels, where the sclerotomal borders meet and stuff, they do not co-localize at all. So I think it's just a technical issue about spillage at a superficial level. Any questions from the Zoom? No?
Hi, one.
Hi, whoever you are.
Sarah.
Oh, okay. Hi, Sarah.
So very nice job. It's really fun to see how this all is finishing up for you. I'm just wondering if you think the second half of your project, if you think that could be extrapolated to neck injuries where like maybe you broke the neck, assuming you're not dealing with like spinal cord injuries, and muscular healing as well. Because a lot of people end up having fusion going on there. Do you think this could be extrapolated to help with regenerative experiments or therapies?
So it's a little hard to hear you. So do you think, are you asking about congenital spinal fusions or?
No, if you had a breakage or an injury.
Oh, if I had a breakage or injury and then that resulted in a fusion, could this help those types of things?
Well, usually what they do, if you have a spinal cord breakage, not spinal cord, a vertebral breakage, like in an accident, you'll fuse those bones together and then you lose mobility. And so, you don't have the muscular mobility, you've got like the lack of the cartilage going on there now, and you just fused it. So I was wondering if you could extrapolate experiments to provide a way to perhaps reform some of those supporting structures.
Yeah, so especially the first project, I really hope so. I really hope that those, especially since it is focused on TGFbeta, that at least even if it's not TGFbeta signaling, the marker stat, have been identified to be so important for that process can be used in a repair therapy, so that in the future, we would have to just take out those tissues and fuse them. That maybe we could apply, some type of ligand or some type of, in vitro therapy before, and like go in there and really target pathways to help repair. So that's the goal, honestly, yes.
Okay.
Yeah, thank you for your question. Cool. Well, thank you guys. This is cool.
So now the committee will